Hi, I'm Leonardo Pellegrina, a postdoc from the University of Padova. In this video, I will present you our work, Discovering Significant Evolutionary Trajectories in Cancer Phylogenies, a joint work with Fabio Vandini. Cancer is an evolutionary process driven by the accumulation of genomic alterations over time, which drive the tumor growth into a collection of clones or sets of cancer cells with the same set of alterations. A critical challenge in precision medicine for cancer is to understand the clonal composition of tumors and how tumors evolve. What is possible today is to infer a phylogeny for each tumor, or a tumor tree, describing the evolution of a single tumor, in which a node is a clone that contains all alterations unique to that clone. This means that a cell that belongs to a clone also carries all alterations found in all the ancestor nodes, as you can see in the example on this slide. However, a critical challenge is to understand if there are conserved progression patterns of alterations shared by many patients, a problem that cannot be addressed by considering each tumor tree individually. For this problem, previous works either focused on partitioning trees into clusters according to their similarity, or to only find simple progression patterns, such as pairs of alterations with a given order or linear paths. So this was the motivation for our algorithm Mastro, for the discovery of conserved and significant progression patterns of alterations. Our algorithm can find trajectories with complex topologies that directly encode co-occurring and clonally exclusive interactions of alterations. It is based on new statistical tests to assess the significance of the order of alterations within trajectories, and on powerful procedures to control false discoveries, providing rigorous guarantees to its output. Okay, so let me describe a bit more formally the computational problem that we want to solve. So first, the alterations that we consider in this work can be any type of somatic alterations of the genome, such as ISMBs or copy number aberrations, and we define a trajectory as a tumor tree that is observed in a patient's tumor tree if all alterations pairwise orderings are the same in the trajectory and in the tumor trees. For example, here we show a trajectory composed by 3 alterations that is observed in all the tumor trees, in which we highlighted in green the nodes in which the alterations are found. And note that alterations are not necessarily consecutive in the tumor trees, but only that their order must be the same. And this is another example of a trajectory that is observed only on the first and the third tree. Furthermore, we only want to consider maximal trajectories, which are the ones with the largest set of alterations that are observed in the same set of tumors, so that they are the most complete as possible. Great, so now we are ready to define the computational problem that we want to solve, that is the frequent maximal trajectories problem, which we are given in input a multiset of n tumor trees that are inferred from sequencing data such as bulk or single cell, and a frequency threshold sigma, and the output is all the maximal trajectories that are observed in at least sigma tumor trees. And here we show an example of a solution of this problem for sigma equal to 2. In the paper, we prove that this problem is NP-hard, so it is unlikely that we are able to solve it efficiently for every instance. So we design a fast algorithm for this problem called Mastro, which the main idea is to expand each tumor tree into a tumor graph, in which we add additional edges to the tree, connecting every node to all other nodes that are reachable from it, as you can see in the example. And the intuition is that a frequent trajectory now corresponds to a frequent set of edges of the expanding tumor graphs. So Mastro finds all frequent trajectories from frequent sets of edges, and can do so by enumerating frequent item sets, a very well-known problem in data mining that can be solved quickly in practice. Now I would like to present you some of the experiments we performed in our paper. We applied Mastro to cancer data from 123 acute myeloid leukemia patients. On this data, Mastro finds 123 maximal trajectories that are observed in at least 2 patients in a few seconds, so it is very fast. It also identifies 40 trajectories as the most significant with small FTR that we show in this figure. Instead, I would like to present you a summary of these trajectories in which we show that they can be composed into 4 major different progression patterns that are observed in different subsets of the patients. Interestingly, we notice that the first 3 patterns are known to be important in AML. In the summaries A and B, a mutation in an epigenetic factor, such as DNMT3A or IDH1 or IDH2, precedes a mutation in the nucleophosmin molecular pathway, NPM1, that is later followed by chronally exclusive alterations of signaling genes, such as RAS and FLT3. While in the summary C, the initiating event is DIT2, another common pattern in AML. Mastro also finds a new progression pattern that was not previously reported, here shown in summary D. This pattern may describe a different modality of evolution, characterizing a subset of patients not hit by an early alteration of an epigenetic factor, in which the first event is a mutation in NPM1, not preceded by any other alteration. So to conclude, Mastro is a fast and effective method to find conserved trajectories of alterations from large tumor cohorts. It is based on new statistical tests to assess the significance of the order of alterations and effective procedures to control false discoveries. In simulations that we show in the paper, Mastro is robust against false discoveries while achieving high statistical power, and on cancer data it allows to discover known and new progression patterns. Finally, I would like to thank my co-author, Fabio Mandini, and say that the implementation of Mastro is available online, and also that our paper will soon appear in Bioinformatics as in the proceedings of ISMB 2022. Thank you and goodbye.